The next structure we're going to be discussing is the cytoplasm. Now the cytoplasm more or less suspends all the organelles within the cell so that it's not all stuck on the bottom. And I'll demonstrate that by using an empty canister. Imagine there was no cytoplasm within the cell and we dropped the organelles into it. As you can see, everything kind of falls to the bottom, which isn't very helpful at all. Boop, boop. But instead, the cell has cytoplasm, which I like to think of as kind of like the cell soup. It contains all the nutrients, the proteins, and the organelles within the cell. We're going to make a model of this by using some of these items. First, you want to find a canister, ideally clear. I found these at Rite Aid in a little kind of travel pouch of soaps and things. But you can use if you have a film canister or any kind of clear container.
With a lid.
With a lid, ideally, yes. You can gather a variety of little arts and crafts kind of objects. You can use buttons, you can use sparkles. Here we have a variety of sparkles and little orbs and whatnot. A toothpick or a popsicle stick to help place the objects into the cell. And of course, the cytoplasm itself. Here we're using some hair gel. You can use any sort of clear or semi-clear gelatin-like fluid. Very viscous, using our vocab words from earlier this year. I guess you want to keep the lid on so you can squirt it in there proper and nice. First we want to just insert the cytoplasm into our cell. Like that. Got some bubbles and things inside there. Next we're going to demonstrate the organelles by picking a variety of little objects here. I'm going to use, I'm first going to insert the nucleus.
And if you're really thinking about it, you can make all of the different organelles different colors. So you can choose all green sequins for the chloroplast, all yellow sequins for the mitochondria. Get creative, but think about it.
Okay, so I'm going to make a plant cell. I'm going to put my green, whatever this thing is, in there as some chloroplasts. And some mitochondria in there.
You can put as many or as little organelles inside as you'd like, but try and get at least one representation of each organelle. So the Golgi body, the chloroplast, the mitochondria, try to get everything in there, at least one example.
So I've got my nucleus, I got my chloroplast, mitochondria, I got my Golgi body in there, my cell membrane and cell walls being demonstrated by the outer side of the container. And when all's said and done, you will put your lid back on there, and you'll have a little example of your cell. Extreme close-up.
Extreme close-up.
Cut.